All right.
Perfect.
Okay, thanks all for giving me the opportunity of talking today about our results, combining experiment and maths to both understand and control complex dynamics in mammalian cells. So in recent years, it has become clear that dynamics in space and time at the intra and intercellular level are key to regulate phenotypes across systems. This is true, for example, in development. Indeed, oscillations of genes sometimes acting to tightly control somatogenesis. This is true also in vitro cultures of stem cells. I will be talking about this in a minute, but also in vitro culture of stem cells, dynamics of genes such as beta-catenin and other genes tightly control the fate of the cells, so controlling the fact that they are more or less pluripotent, basically. This is true also in cancer. Here there is an example of a signaling pathway for which it has been reported that providing different inputs upstream of the pathway, different dynamics happen in normal versus cancer cells. So again, there is a link between dynamics and phenotypes. And for sure, this is true also in tissues, where also intracellular dynamics matter. For example, how cells move, the forces among cells, and the cell cycle. So what my group is trying to do, combining system and synthetic bio approaches, is both trying to understand how collective nonlinear dynamics are achieved. We are also trying to understand if and how these dynamics affect cellular phenotypes. And finally, we are trying to directly control these dynamics, combining control engineering with synthetic bio. I will start with some systems bio results. In the lab, we also perform experiments, and we use mouse embryonic stem cells as an in vitro system to study development. Briefly, mouse embryonic stem cells can be isolating during in vivo development from mice. The stage of isolation matters, so if cells are isolated earlier during development, they will be more pluripotent as compared to cells isolated later. We work with mouse embryonic stem cells, so they're supposed to be the most pluripotent ones. Still, the medium in which we culture the cells matter. Serum leaf is a media which keeps them between a more naive and a more primed for differentiation stage. While to leaf, I will show you experiments and models of cells cultured into a leaf, is a more recently proposed media which is supposed to keep them more in a ground state of pluripotency. So a few years ago now, we started to look at beta-catenin, which is a key regulator of pluripotency. Briefly, we performed experiments by tagging the endogenous gene with the GFP, so with the green fluorescence protein, in such a way that we could look at the cells at the microscope. And what we noticed in, again, mouse embryonic stem cells cultured in serum leaf was the fact that beta-catenin, here we're using the fluorescence reporter as a proxy, was heterogeneous, with some cells expressing more and cells expressing less beta-catenin. This was happening actually also for Nanog, a master pluripotency regulator already reported previously to be bistable. And also when we looked at thousands of cells by flow cytometry, we noticed the presence of a low population and a high population of beta-catenin expression as compared to negative control. Not showing this here, but we also performed time lapses where we observed that cells with low beta-catenin were able to switch it on and vice versa. To make sense of this heterogeneity and fluctuations in time, we developed a model describing this gene regulatory network. In this network, the standard error represents activations and the blunt errors represent inhibitions. We actually firstly proved this new interaction with experiments, and then we developed this model. So here we have a function, so it's a fully nonlinear model to represent activation and inhibitions, and also we have got some white noise. So these Gaussian terms here are representing stochasticity. Combining literature parameters and parameters we directly measure in cells, we were able to recapitulate for both these proteins, so for both Nanog and beta-catenin, a bimodal distribution at steady state, which was matched to experimental data for both proteins. And then we also performed a bifurcation analysis that basically allowed us to at least suppose that the system is bistable. So looks like for both proteins, there is the coexistence of two stable solutions for both Nanog and beta-catenin, separated from an unstable one by two saddle node bifurcations. Then that we wanted to look more broadly at what happens when you shift cells from serum leaf media to a leaf, which is again, this media that should keep cells more pluripotent. And we wanted to recapitulate experimental data reported in the literature about the fact that, again, Nanog, this key regulator of pluripotency, is bistable in serum leaf, as you can see here, at steady state from flow cytometry data. In leaf, as supposed, the cells are more pluripotent, so we have more cells expressing Nanog high, but still there is a small proportion of cells with Nanog low. This was observed both at steady state and in time lapses where cells were fluctuating between the two states. And we wanted to understand from a mathematical point of view why this was happening. So the first thing that Simon, a former PhD student in my group, did was to derive this gene regulatory network for stem cell pluripotency. Briefly, he combined RNA-seq and CHIP-seq data previously published. And the network contains key regulator of pluripotency as well as PD and chiron, which are basically the inputs to our systems because they are the two drugs contained in the 12 media. And it encompasses both transcriptional interactions, are these solid arrows, and protein-protein interaction. Again, from the literature are these dashed lines. This model is similar to the one I showed you before. It's just in a higher dimension. So again, we have got L functions, and again, we have got Gaussian terms to describe noise. Something else that we wanted to recapitulate was not just the distribution of Nanog in the two media, but also different proliferation dynamics that we and others measured when cells were cultured in the two media. So what we did was also to extend this model to an agent-based version using CHASTE, that is a C++ framework to simulate mammalian cell agent-based multiscale dynamics. So each cell again contains this stochastic differential equation model to represent interactions among genes. Also, we coupled this network to the cell cycle. Specifically, we coupled this gene MECAN to the cell cycle by basically assigning to cells with low MECAN levels a longer cell cycle, so slower proliferation as compared to cells with high MECAN. And finally, we are also representing forces among cells. This is a quite standard Voronoi tessellation representation. This model was able to recapitulate quite well, we think, although there are lots of parameters there, so we couldn't measure all of them directly, but it was able to recapitulate quite well steady state dynamics of Nanog, both in serum leaf, this is the model, the blue line is the data, and in leaf. So in leaf, also in the model, we still have a few cells with Nanog low. It was validated against a different experimental data set published in the literature, in which cells were sorted for Nanog high on low, and then they were measured in time. Data and experiment, there is a decent match, we think. And importantly, the model was able to tell us why we think that in leaf, not all the cells express high level of Nanog. So what we think is happening is that, again, in serum leaf, the system is bistable. In leaf, we move towards the high Nanog steady state, but we think to still be very close to the saddle-node bifurcation, meaning that being the system stochastic, if the noise is high enough, some cells can escape the Nanog high state and still basically move here. And this should explain the presence of this Nanog low population both in the model and in the data. To conclude this first part of the talk about systems bio results, we are using agent-based models also for tissues. In particular, we are looking at intestinal crypts that are glands lining both the small and the large intestine. And they are across the organs that renew the most in the organism. That's due to the fact that at the base of the crypts, there are stem cells which can proliferate quickly and are pluripotent, but they differentiate as they move upwards in the crypt, becoming differentiated cells with slower cell cycle. This phenotype is due both to the position of cells, but also to the fact that there are a number of signaling pathways with gradients across the crypt controlling the fate of these cells. What Daniel, former PhD in my group, did was to develop, again, an agent-based model in CHASTE. We simplified the geometry to a 2D geometry, so basically like an unrolled cylinder. We can represent different cells into the crypt, we can represent how they move, and also we can reproduce gradients of signaling pathways. For example, again, the Wnt pathway, which should mimic both small and large intestine dynamics. And also within each agent, we can represent, again, with differential equation, dynamics of key signaling pathways. In particular, we represented the Wnt pathway. and its coupling to both the cell cycle and in this case also to adhesion among cells properties. And we coupled the Wnt pathway to the hyposignaling pathway, which is a pathway recently reported to be very important for regulating cancerogenesis in intestinal crypts and also in organoids. Not showing all the question because this is a very complex system. I just want to give you an idea of the kind of simulations that we performed. So in this video, I'm showing a simulation in which we tested a novel hypothesis from the Hans Clever lab, in which basically they hypothesized that even without imposing a gradient of Wnt in the crypt, if cells at the base of the crypt stem cell express Wnt, it should be able just by dilution as they migrate upwards for them to basically, as they divide, to dilute Wnt, so to create the gradient. And actually this is the case, as we tested with the model, both in absence and in presence of noise at the time of cell division. So in this first part, I showed you a combination of experiments and maths to measure and explain nonlinear dynamics in stem cells and in intestinal crypts. We were quite happy with those results, although we know that given, especially with the intestinal crypts model, the complexity of these formalisms, the number of parameters is really high. So not always we are 100% sure that they are unique to describe our system. Also, we measured, for example, for beta-catenin this by stability, but we don't have so far a way to directly link these dynamics to phenotypes in stem cells. And for these aims, we are also using synthetic biology approaches in which instead of measuring what happens, we directly engineer specific gene expression dynamics to then couple them to cell behaviors. And to do this, we exploit feedback control. So there are mainly two approaches in synthetic bio. Either you can have both the controlled processes and the controller inside the cells, so implemented via biomolecules, or the controlled processes can be into the cells and the controller can be externally implemented through, for example, a computer. And this is what is called in silico external feedback control. So just to give you an idea of the kind of experiments and maths that we are using, in terms of embedded control, although actually this system is in open loop, we developed in the lab and we recently published in this paper a synthetic tool that allows us to control for an exogenous genes both transcription and protein stabilities with two orthogonal drugs. Without going too much into details, they're called DOXY and TMP. We measure the fact that actually these drugs are orthogonal, and that's the case. So one just control transcription, this is the qPCR data, and the other TMP just control protein stability, as that's by flow cytometry. We also developed actually in this case a differential equation without noise model of the system, and we proved that it allows a wider dynamic range as compared to system in which you just control transcription on translation. And then we started to test how doses of genes, such as again beta-catenin, affect phenotypes, and this seems to be a link. This is called an APC staining. Basically, the clones that are more red, more stained, are the ones that are more pluripotent. And there are some media conditions for which lower doses of our exogenous protein allow to have more pluripotent colonies as compared to higher doses, meaning that it seems to be a robust tool to again associate doses to phenotypes. Also, we are applying what I mentioned earlier as external feedback control. So what we have got here is a standard cross feedback strategy in engineering. And this is our platform to basically implement this for living cells. So what we do is to culture cells in PDMS microfluidic devices that we design and build in Bristol. And as we culture them, we perform time-lapse imaging. Each time we acquire an image, we run online segmentation algorithms. So far, there were simple segmentation algorithms based on thresholding methods. And each time we compute the output, basically is mainly so far based on fluorescence reporters. It is compared to our control reference. This allows us to compute a control error, which in turn informs a control algorithm, which automatically changes the media that is provided to cells in such a way that hopefully the control error is minimized. So this slides some further details about our experimental platform. So this is again our chip. These are the chambers in which we grow our cells. The chip is placed on the stage of our wide field microscope, and in turn, through these lines is connected to our syringes, which are automatically controlled by MATLAB that online performs both segmentation and control algorithms implementation. I would like to show you a couple of examples of these experiments. So in this case, this is again our system that allows us to control both transcription and translation. And we are trying to control an exogenous fluorescence protein. In this case, is an mCherry. And in this plot in blue, we have got the control reference. In black, the control inputs, using here a very simple relay control strategies, which still is able to force the measured fluorescence, the red line, so the control output, to track this time-varying control reference. On the right is very similar, again, a relay control time-lapse experiment, but here we are not just controlling the mCherry, but also a gene of interest, which is fused to it. And in this specific case, the control reference is a bit more simple. As you can see here, is a set point control reference, which is equal to 50% of the maximum activation of our exogenous gene. We are also now trying to implement more sophisticated control strategies, such as model predictive control. They require a model of the system, but what we have found, at least for the system we analyzed so far, is that sometimes simple model can be effective. In this case, for example, again, this is the system to regulate an exogenous mCherry fluorescence protein. What we did was to perform firstly some open loop experiments in which we provide to cells a priori determined inputs, so basically a combination of the two drugs, dox and TMP, that activates our output. We measure the output, and then we were fitting some very simple linear state space systems, which gave a decent fitting that actually was also good enough at the time of performing on the same cells which are embedded in this construct, model predictive control, again with the set point reference. So now that we have got this platform that allows us to control mammalian cells, how are we going to use it? We have got mainly three aims. This is work in progress. The first aim is to try to implement control-based continuation on biological systems. What's the idea of CPC is basically to skip this step. So what I showed you before, we perform experiments, we derive the model, and then we did bifurcation analysis to extract nonlinear features from the model. But what if we don't have a model available, or we are not 100% sure about the model? CPC, what it tries to do is to directly extract those features from an experiment. And the way it works, here there are a few very good references from the literature, including works from Dave Barton, a colleague in ENGMATS, that has been pioneering these techniques. So the idea is that by applying feedback control, if you are unable to apply control that is non-invasive, and if you are able to stabilize all the solutions, also the unstable one, you should be able, in principle, to offline reconstruct the entire bifurcation curve without a model if you use a control strategy that is model-free. We didn't do experiments yet, but we are testing at the moment this in silicon. So again, using the model I showed you before about Nanog, what we did was to simulate the system in open loop first. And as you can guess, even if the system is best stable, if you simulate it in open loop, you can't observe the unstable solution. So observing your output Nanog as changing the bifurcation parameter, that in this case is also the control input, they are supposed to be the same. You could observe just the two stable solutions. While, at least in silico, applying CPC, if we properly design a control reference as a step function, we should be in principle able to stabilize also the unstable solution in such a way that offline we should be able to reconstruct the full bifurcation curve. We didn't perform CPC experiments yet, but what we are starting to do at the minute is to control mouse embryonic stem cells for the expression of pluripotency genes, and it seems to be feasible. In this case, for sure, the control reference is much more simple. It's that point. We need to find ways to implement the full CPC. And a big issue are delays. It takes quite a lot of time to stabilize mammalian cells, so we might think also about different platforms, not microfluidics, to perform longer experiments. On those lines, we are also planning to basically use the platform to automatically infer optimal protocols to culture stem cells for a long time. And we are planning to do this across scales from microfluidics to bioreactors. And the idea would be both to compare different culture conditions at the end of the experiments and to use agent-based modeling to compare different dynamics that we expect to see when scaling up from microfluidics to bioreactors. And finally, within the aims of a project funded by PSRC with AstraZeneca on board, is to use the platform to automatically design superior treatments for cancer. So platform the same, difference is that we will use cancer cells chemotherapeutic agents instead of drugs that perturb, for example, stem cells. And as control outputs, we will look at reporters for cancer, for pathways which are misregulating in cancers all the cell cycle. And the idea is to start in open loop. So again, with inputs that are determined a priori, measuring the output and deriving black, as in the example I showed you before, or gray box models. And then the idea is to close the loop. So basically setting the control reference, which could be, for example, expression of specific genes in pathways misregulated in cancer, and letting the control algorithm telling us which is the best combination of drugs to have that desired output. We didn't start doing closed loop experiments yet, but what we're doing at the moment is to start culturing non-small cell lung cancer cells in our chips, and they seem to survive. We are also engineering them with reporters that basically will be our control outputs, for example, reporters for the cell cycle. And in the meanwhile, this is a working collaboration with Tom Gorochowski in Bristol. We are improving our computational tools. For example, we are developing segmentation softwares that are based on deep learning using UNet architectures. With this, I want to thank all the members of my group that were generating the data I showed you, all the founders, the collaborators, and all of you for your attention. Thank you.
Thank you very much, Lucia. We already have two questions for you. The first one is there a specific reason of using multiplicative noise in the model?
No, there is not. We did this because, let me put the questions on. We did this basically because in particular for this system, we started from a previously published model. And the reason for doing that is that, as you can see, the number of parameters is quite high here, and we couldn't measure all of them. And the structure of that model was something like this, and we kept it the same in such a way that then we could also adapt the parameters that fitted previously for our aims. That's the key reason.
Yeah, thanks. Great. Next question is, so has all the feedback control you've used based on fitted linear models? Do some of your control strategy utilize fully nonlinear models you described earlier in your talk?
Yeah, so it depends. So far we are trying to keep things simple as far as they work. So for example, in these two experiments that I showed in this slide, it was completely model free. It was relay control. And also for CBC, so in this example here, I didn't mention it, but the controller that we used is a simple proportional controller. And the idea is that as we want CBC to extract those nonlinear features without knowing the system, we want model-free strategies. In terms of model predicted control, in the example I showed you, the model was linear because linear was working, decently. But for other systems, we are using gray box approaches. There are other systems for which, for example, we imposed a nil function because we know that it's important to include their saturation and nonlinearity. And so it's more a kind of a gray nonlinear approach and is a nonlinear one. For the cancer cells, I don't know yet which kind of approach we will use. It might be that we need a more sophisticated model also because for two reasons. One is that we are controlling endogenous processes as compared to the exogenous processes we have been controlling so far. And we know that they're much more nonlinear and there are big cascades there, and we might need to capture that complexity. And also because we might go for multiple input control. So we might need to have more sophisticated models, but at the moment, I don't know.
Thanks. And one more very interesting question from Kyle. Thanks for the great talk, Lucia. At the beginning of your talk, you discussed the large degree of heterogeneity in cell states, like the cell state marker, like Nanog. For your control strategy, are you using fluorescence signals for populations of cells or just a selected few? And if the former, what is the efficacy of the control strategy for heterogeneous populations of cells?
Yeah, thanks for the question. So at the moment, the experiments in this slide, basically I was describing the experiments that we are performing to control pluripotency. We are using a cell line that was established by another group in which basically, yeah, they're tagging with the GFP, REX1, the endogenous REX1. So it's a quite good reporter. In terms of the kind of control we are using, so far in the majority of the experiments, we are controlling the population, also because very often these cells grow as big clones. So we can't, when we segment the image, the part of the chip, basically isolate individual cells. So it's a population control. Nevertheless, at the moment, we are basically designing and building a new chip that should allow us to grow the cells as more flat cells. And we are also trying, from an experimental point of view, different kind of coatings of the chip that should allow the cells to be more flat. I'm saying that because if we can do that, we should also be able to count the cells. So not just having a big clone with lots of cells there, which in turn should allow us to control the distribution of cells. And that would be much better. Because at the moment, by controlling the average in the population, we are not sure about what each cell is doing. But we have got a few experimental issues to sort out first and segmentation issue to do that.
Yes, great. Thank you very much.